Okay, so welcome back, everyone. So today it's my pleasure to welcome Lin Wan, who will be talking about learning complex cellular dynamics from time series. Yeah, thank you.
Okay, thank you for your invitation, and I'm very happy to present some work on learning complex cellular dynamics from time series sequence data through our developed integratable generative diffusion model. So, okay, sorry. So from this, if you look at this picture, you will understand it is a statistic picture, but you can see that there's a rich dynamics inside this picture. Can we predict the dynamic of this from the picture? I think everybody can predict, even the no need of physics. But can machine predict the dynamic of the picture? I think it's very quite a challenge for machine to do this. Recently, generative AI has played a powerful role in generating different kind of things, such as video, text, but also recently, the generative AI has been used to generate videos. But this video, although this video looks most realistic looking, but they can still be and criticized. For example, some people say that the generation is different from the causal prediction from a word model. And our question is, can we, the generative AI, really understanding the physical world? So how can we improve this kind of situation? So in this talk, talk about our recent work on learning dynamics from time series snapshot data. Its data is different from traditional time series data. It's a collection of cells or ensemble of cells from samples at a different time. So we need to develop a new method to understand this kind of data. This data, there are some methods have been developed using optimal transport to analyze this kind of data, but this kind of data, this method can have different pros and cons. Here, I will report our two works to understand this, to learning the dynamics from time series snapshot data. It's one work is called GraphFP, and the other work is called PySDE. I will give the details, and finally, I will give a summary and future work. So I give a little background. As everybody knows, that a cell is the basic unit of our human or animals, and different part of different cells is not different. It can be different in different tissues. The cell can be totally different to perform different functions, and to underline this biological system, there's a dynamic system there where the cells can grow from the stem cell, which is have multipotence to, and a very complex development process can develop into different kind of cells to perform different functions. So can we understand this underlying dynamics is very important for the life science or even for the medicine. And also for the tumor, for the cancer, there are also some complex dynamics underlying these cell processes. So can we understand this process? Recently, the technique has developed. There's called a kind of machine is called a single cell sequence technology. They can isolate the cell from the tissue and measure the genes inside the cell. So the data is usually looks as like a matrix. For each row present a different cell. For each column present different genes. The expression of the genes. You can think of this column as a feature. So this data is a very high dimension. But the problem for this data is that the experiment is destructive. After you sequence the cell will be killed. So a lot of information. You cannot sequence the cell for many times. You can only sequence it one times. So for this reason, the problem is that for this reason, each cell can be cell one times. So when we sequence the tissue or a sample from different time point, we cannot trace each individual cells along the time. But at each time, we can only sample a batch of cells from the population. So the problem is that at each time point is a statistical snapshot, which means the data are sampled at maybe irregular and sparse time point. And for each cell, it's a high dimensional data of the cell states. It can be 10,000, more than 10,000. And because of the destructive nature of the cell, we cannot trace each individual, and we call this kind of data are unpaired cell. You cannot match which cell to the next cell in the next time. And also the cells are very heterogeneous. And during the process, the cell can have interaction or communication during this process. So our problem is that given this kind of time series snapshot data, can we learn some of the dynamics from this kind of data? It's more like if you have a movie, you can have you only can know some images at a certain time. The problem is, can we reconstruct the whole movie and can also predict the future, predict some unseen part of this data. Also, can we quantify how this characterized in the movie their relationship? So for our problem is more than that. We have the snapshot time series data. We want to align this unpaired cell across time, and we want to reconstruct the potential energy landscape that characterizes the global picture or global dynamic of the system. And we also inference or predict each cell velocity, or we want to understand at each local position how this cell evolve or their dynamics. Also, can we understand their cell interaction from this kind of data? This is the big picture of what we want to do. But the problem is very challenging because this problem we cannot trace it in individual cell. At each time point, we only have very high dimension distribution. So the problem is that for here is rho, we can think it's a high dimensional distribution or density at time t. The problem is that can we found a path that connected different this distribution in the probability measure space that fit the data distribution. If we have some model for this kind of data. For example, if the cells are governed by the stochastic diffusion equation or diffusion process, this model, this equation should be Fokker-Planck equation. But how do we fit the Fokker-Planck equation in this kind of data? This is basically the problem. Many authors have realized this kind of research. For example, George Kanadikas, they recently published a paper that they call this kind of research as ensemble regression, which is different from traditional point regression. Traditionally, we have at each time point we have one data point, and you want to fit the curve along this point. But currently, at each data point, we have the distribution is kind of a problem measure space, and so for the point regression, you usually work on the Euclidean spaces, and your goal is to fit each individual point, and the metrics here can be Euclidean distance. But for the ensemble regression, at each data point, you have a probability measure, and you wanted to fit the particle ensemble or fit the distribution. So to fit this, to measure to measure this distribution, here we use some other metrics such as Wasserstein distance that I will talk soon. This is basically the problem. So it's how do we fit the distribution along the time? So the major tool that can be used to fit the distribution or how to measure the distance between distribution is called optimal transportation, optimal transport. Here we use the form that's called Mongo-Kontonovich optimal transport plan. Here, for given cost function C, we wanted to transform the two distribution mu and nu. We wanted to transform the transform the distribution from mu to nu with a minimal cost. Here, the pi we want to find a joint distribution of x and y, which transfer mu into nu with a minimal total cost. We call this pi is called probabilistic coupling or alignment. Here, if when the cost is the Euclidean distance between the point x and y, this kind of measure is called Wasserstein distance. It measures the distance between two distributions. So with this kind of framework, we can deal with the distribution problem. So for the first method to modeling the time series snapshot data for cell is called Wellington OT. It's published at 2019 in a Cell paper. Basically, it's a classical optimal transport problem. It calculates the OT problem or optimal transport between each adjacent time point and get the temporal coupling. With the probabilistic temporal coupling, you can inference the cell development trajectories based on this kind of temporal coupling information. You can get a probabilistic flow along this time, but basically it's a statistical mapping. It's you cannot use this to make a prediction. So another method is called other researchers use the dynamic OT. It's a kind of a dynamic view of optimal transportation. This form is called Binomos-Bruni formula. It formula the OT problem into the you wanted to find a vector field V. With this way you can transfer this mu to nu with the minimal transportation cost. And the problem is that we wanted to find the vector field V that transport the initial target distance density mu to the target distance to nu with the minimum total kinetic energy. This is basically the model cells, and the V and density rules should satisfy the continual equation, and with the fixed initial and final time point condition. So this kind of problem is a kind of optimal control problem, and also we can call this kind of it's kind of a principal selection strategy to regard this kind of problem. So the translate is a kind of method that developed based on the Binomos-Bruni framework. But here it combines the dynamic OT with the neural network called continuous normalizing flow. They use neural network to try to model this vector field f theta to learn this from the neural network. And here is basically the formula. They want to find a neural network that can with a minimal kinetic energy, and also they also want this observed data and the model predicted data have a very small distance of chiral divergence. And here I want to emphasize that this kind of dynamic OT problem is kind of deterministic model. It's OD. It's kind of ODE. Here the X follows the OD. And another method is called Wellington. Is kind of another one other method wanted to reconstruct the underlying landscape of the dynamic. Here is called a method. It's called it's a theory called a metaphor theory called Wellington landscape. It's developed in the 1940s. It says that if the cell has polyphony potential cells, it should have a high potential energy, and it's more like a high potential ball rolling down along the hill to different the valley. At a different valley, it's called a kind of cell states or cell fate. And here many methods want to reconstruct this kind of landscape, and some methods use to reconstruct this landscape from the data. We call this phi, and we think the dynamic of vector field is induced by the potential energy landscape and the gradient along this kind of potential. So the present is the method that is kind of basic kind of things to want to model this kind of landscape. It's kind of a stochastic diffusion equation model. It's a generative diffusion model, and the model for this SDE, the drift term is modeled by the gradient of the potential. And here is the Brownian motion, the Brownian motion part. And given the initial distribution of the data can generate the development of this dynamic. Here to fit this kind of data, this method also want to use the neural network as a surrogate model of the phi. They want to use neural network to learn this phi. So this is data first they fit they wanted to first the loss function of this method contain consistent of two parts. The first part is called Nist-Wasserton distance. They wanted the observed distribution of the data can have is similar to the predicted distribution by the SDE. So they want to minimize this distance. Also, this also think that the cells should have a low dimension when they developed. So they have a regulation term that penalizes that cells penalizes the potential of cells had at the final time point. So that's what we call it. It's a local regulation term, because this regulation term only functions on the data collected at the final time point. So we have found that this kind of regulation term is over reliance on the training data. So it's like interpretability. I'll show you the result data. So before I go down, I give a summary. So there's some kind of a statistical optimal OT transporting-based method, such as Wellington OT. This kind of method cannot predict unseen data. For the dynamic based OT-based method, such as trajectory net, it's kind of deterministic data. But this data, we found that for the deterministic model, you have a lower accuracy in prediction. This kind of data also have a mechanism-based that uses the principle of least action in their model. But this method cannot provide the potential. For the diffusion-based method for the SDE, it's a stochastic model. It's a stochastic model. It's a diffusion model. It can provide potential. Most of the method is fit the predicted and observed data using Nist-Wasserton distance. But here they use the empirical local regulation at the final time, and there is no mechanism assumed during this machine learning process. So to our problem, we want to learn the cell dynamic using the diffusion model, and we also want to use this generative diffusion model such as the stochastic diffusion equation. But here the difference from our method and the preset is that we want to introduce some mechanism into this machine learning method. We call it such as the principle of least action, which has been widely used in modeling of biology and physics field. And for example, we wanted to minimize the total kinetic energy as the dynamic OT did. But if we want to impose this kind of mechanism into the system, we are changing because this kind of is SDE problem, we will change the problem into the stochastic problem, control problem, or reinforcement learning problem. So analytically, to solve this kind of stochastic problem, usually we should work on the forward-backward partial diffusion equation, which for the forward part, it's a Fock-Planck equation to describe the development of the SDE, and for the backward component, it's kind of a Hamilton-Jacobi equation. It's from dynamic programming, this kind of equation. But this equation is high-dimensional PDE, so it will be due to the it's a computational dotting task because the curse of dimensionality. So the problem is we face that if we want to impose the principle of least action in the SDE model, the competition is a big challenge for us. So to deal with this problem, here we have two strategies to deal with this problem. We want to use the diffusion model. We want to combine the principle of least action, and we want to use some machine learning tools to analyze this kind of problem. Here, for the first our work, we call it graph FP, which is a nonlinear Fock-Planck equation model on the graph. It's a causal graining method, which is worked as a cell type level, or also called cluster level. The idea is that we aggregate this continuous cell states by clustering or annotation into the finite discrete states or discrete cell type states. So in this way, we can transform the PDEs to the ODEs. That will be very easy, relatively easy to solve. And based on this model, we also can quantify cell-cell interaction and communications. Another approach is we use some physics-informed approach. It's kind of a fine-graining method. It can work at cell or particle or agent level. We want to encode this physical knowledge or principles into the neural network. Here we want to encode the principle of least action into the architecture or loss function of the neural network that makes this method can computationally efficient. So both of methods are computationally very efficient, and most importantly, they can improve the interpretability and prediction accuracy. So for then I'll give more details about these two work. For the first work, it's called graph FP. It's work on at the cell type. It's modeling cell type dynamics. It works as a discrete state space. The idea, as I just mentioned, is that since we can clustering these cells into finite discrete states, and in this case, the group cell into finite states, and each state we note it is as a node on the graph, and if there is a transition between the graph between the cell states, we add an edge between the graph, and basically, and if it's finite at this time point, we can count the frequency of each frequency of the each cell type and get a probability simplex. And at each time we get a frequency. Each setup is at this time point. And we want to model the dynamics of the probability simplex along the time. Here we want to using the diffusion model to model this kind of process for the continuous condition. There's a connection between SDE and the Fock-Planck equation, also with the optimal transportation. For the continuous case, it's called a JKO scheme. It says that the Fock-Planck equation, or SDE, can be viewed as the gradient flow of the free energy functional F. Here, the gradient flow is on the Wasserstein manifold or Wasserstein metric. It's different from Euclidean space. And here, the functional energy F is usually contains two parts. The first part is the potential energy part to characterize the potential energy of each X. And here is the entropy part, is characterize the random noise. Here, our model, because since we can group the cell into the discrete space, we work on the graphs. We work on the discrete space. We call it the graph. We call it the nonlinear Fock-Planck equation on graph. We model here we model the functional of free energy into three parts. The first part is the linear potential part that called linear potential energy. We also want to model how this cell interact with each other. We add another conjugate term to characterize how different states interact with each other. Here we use W to model this, and this term is also a Boltzmann entropy term to model the stochastic part. And for the discrete part, we can parameterize this function into with phi and W. Phi is for the linear potential energy, and W is the interaction potential energy of these different components. Here, N is the number of the states or graph loads. And here W, we do not assume it's asymmetric. It can be a symmetric condition. And so with this setting, so which is this kind of equation looks like. This work is based on the series work did by the Xiu Chou, Hao Ming Zhou, and Wu Chen Li in the recent years. Then they derived this Fock-Planck equation on graph as the following ODEs, and it's kind of ODE since it's a finite space. It's kind of ODE, and here F is we also kind of call it it's kind of dynamic potential. It contains three parts. It's the linear part and the interaction part for the Wij, and the basic idea is that if the cell or states have a high potential, tends to transition from high potential cells to the low potential cells. This is basically the framework, and to impose the least principal least action or minimize total kinetic energy. Here we wanted to minimize the vector the total energy with this term, and we also want to minimize the observed distribution and the predicted distribution by this ODE. And so this part contains two parts: the principal the principal least action part and this part. And as you will see that because this equation is ODE, and its kind of problem is kind of optimal control problem with the fixed initial point. And for this ODE problem, it can be easily solved by the Pontryagin maximum principle, or also called the adjoint method, which has been implemented by the NeuroODE in recent years. So with this kind of framework, we can easily solve this problem by the NeuroODE, and we can estimate this parameters such as phi and w based on these settings. And because the potential energy here, they have different meanings. For example, the phi, we can use this, I'll show that we can use it to quantify cell diffusion potence. And for the interaction terms, we can use it to derive cell-cell interaction from this term. And here, this entropy term to measure the stochasticity of the dynamics. And for Wij can either greater than 0 or less than 0, they can have a different underlying biological meaning. And here's the flow chart of this work. Basically, we have the data. You cluster the data into you cluster the cell into clusters and count the probability frequency of each cluster, and put it into our model, and we can first estimate these parameters such as potentials and interaction terms. And once we learn this method, we can use the method to quantify the probability flow along the time to predict the dynamics over the time, and also to quantify the dynamic potential of the cells. So here is an example. We work on the neural cell dynamic process. It's kind of data collected at different time point and these cells some cells are in their earlier precursor stage, some cells are intermediate development cells, and some cells are different cells such as young cells, neural cells. Our method first can quantify the state transition during the development with you can see you can find that in the early stage these AP cells or RP cells primarily transition to the young cells and the microneural cells. In the later stage, this IP cell intermediate cells will transition to the macro cells. It's basically consistent with the biology. And we also can quantify this potential of this cell type. For example, we found that our estimated potential, we found that the ancestral cell types have higher potential energy, and the decentrated cell types have lower potential energy, which is consistent with the Wellington landscape with the model. And we also can quantify how this is different cell type interaction with each other. For example, if this for this cell type 5, you can find that the interaction term is close to 0. So their dynamic is almost is almost constant along the time. But if you look at this, if you look at these cells that have high interaction value, we can find that if they can play a key role in the development process to, for example, to facilitate such as the transition between cells or prohibit the transition between other kind of cells. So it's give us a understanding how this cell interaction during this process. Also, we want before I close this part, I want to mention that the interaction in our method is very important. Here we check if the model with and without the interaction term, we can find that for the solid lines indicates the method it's kind of it's kind of we conduct the head-out test at each time. we use the red point as a text as a learning sample, and using this blue point as the testing sample. We test the sample with either with intelligent or without intelligent to test whether which model, which method can predict the dynamics. We found that with the intelligent, we can better capture the dynamic nonlinear trajectory of the cell development. And if you have more time point, we have better performance. So this result indicates the integration is important for this process. So that's basically the what graph FPD. Here the second part of our work is called PySDE. It's a kind of physics informed SDE model. Because for the graph FD, it's only can look at the cell type level. It's a causal graining method. We wanted to some fine graining method. Here so how can we encode the principle of least action or minimal action path into our model? If we and we wanted to involve too much computation, here is the solution or method. We wanted to encode our understanding of physical principle into the regularization term that to enforce the system to try to follow this physical principle. So the problem is that how do we design a regulation term to encode the physical mechanism globally? Not that's basically the problem we want to solve. And this term is as precise. That is least Waterton distance between predict and the observed data and the predicted distribution. So here we based on some physical study or understanding, if the system is principle of least action, as we mentioned that it's from stochastic control theory, we can we know that this phi should follow the Hamilton-Jacobi Bellman equation. Here phi is equivalent to potential phi is equivalent to also called value function in the stochastic control area or reinforcement learning area. And another part is from the mathematical biology or from the also from physics area. They call this kind of they have a huge study to call this kind of potential as the quasar potential. This quasar potential is used to characterize global characterization of the transition between fixed point or basin in the dynamic system. And if the system is with weak noise and in the content with SDE with weak noise, this study is mathematical study found that they also will follow the Hamilton-Jacobi equation. It's equal, and from large division theory. So that means if we have some knowledge about this phi, it should be follow the HJB equation or Hamilton-Jacobi equation or HJB equation. So here we wanted to introduce this equation into our loss function. Here we took the reduced form from this study of this Hamilton-Jacobi equation. But solving this equation is still a very challenging problem because it's completely it's a PDE. Therefore, to solve this problem, we follow strategy by Rousseau and Ockham. We relax the constraint of HJB equation by introduce a regularization term that we want to penalize that the violations of the HJB or HJB equation along the trajectory in this form. And we introduce this regulation term into the loss function. And it's kind of, we call it the loss function PI, called physics informed, into this model. But there is still a problem if we, even with this regulation term, we relax this term, but it's still hard to solve because if we want to integration this part, it's a high dimension, and the integration is at each time point is still very computation daunting task. So the problem is, can we remove the effect of the rho t rho tx, and the strategies here we take a strategy as the MCMC scheme. Instead of integral over all the entire space at each time point, we sample at the initial point from the observed data, sample initial point, and generate a different trajectory, and compute the integrals on each trajectory, and we compute the averaged or averaged the loss along on the sampled trajectory. So within this framework, this problem can be solved by the NeuralSD solver. So that's basically the framework. So we want we use the neural network to study this to reconstruct the file. So this is a flowchart of a method. The method is basically, so we have the observed data at a different time point, different distribution point, and we're using the SDE model. And here to learn this file, we introduce some HJB regularization or HJ regularization, and we solve this problem with a neural network. Once we learn this network, we can application this data to many applications. For example, we can use this generation model to predict the unseen time point, and also we can reconstruct the landscape, potential landscape to depict the global dynamic of the system. Also, at a local time point, we can inference this cellular or cell dynamics at a different point to understand these details of the dynamics. I'll talk about some data we tried. The first data is human beta cell development data. This data is collected at 8 days from day 0 to day 7, and it's about totally 50,000 cells. The data are collected. You can see, when we project this data into the, you can see how this data from this, the gray area shows the total data, and the black point shows the data of day 1, day 2, day 7. So you can show the distribution of this data along the time point, and we try our method on this kind of data. First we conduct a head-out task. The idea is that for example, we head out one time point from the data, and use the rest of the data to train this model, and then predict the head-out time point to check the distance between the observed one and the model predicted one. And we compared with their comparative method. We found that our PySDE always have lowest distance, so it have best performance in all the head-out point, and both in the training data and the testing data. So it shows that our model has a very high prediction, accuracy. Also, we wanted to see whether our potential energy learned is biological interpretable. So if we want to check whether it can be consistent with this the Wellington landscape, if that the cell have high in the early stage time point, the cell usually should have a higher potential. If the cell are differential at a later time point, you have low potential. We found that globally that this potential this cell potential will decrease along the time, and if we look at the detailed landscape. Here is a detailed landscape or our reconstructed landscape. For example, here the left one shows the original data, the experimental data, and this blue point show this data cell from day 7, from the final data point. And for this one, you can see that it's the landscape reconstructed by present. We can find that the present landscape is overly dominated by the data collected at the final time. If you look at the final time point, there's a sudden drop at the boundary between day 7 and the other data point. And if you look at this part, this is another branch after the cell development. Usually, this part should have biologically it should have a lower free energy, but due to this point has some data coming from non-day 7 data. This part can have a very high potential, which is not correct. But if we look at the free energy constructed by pi-SDE, we found that the landscape is a progressively decline trend, and it's not affected by the final time point. And if you look at this part, at this branch, it should have a lower potential. It's also our method can predict a lower potential, which is consistent with the understanding of biology. Also, we can the inference cell velocity is a gradient of the potential energy. It's also our method is also can give you also a biological consistent result. For example, for this data, you have two major branching or the branch process at one event is at this here. Branch 1, the cell can develop into two separate parts. But if you count the direction of the velocity for trajectory net, all the directions are to the one direction, and for the present, most of the cells are along the another direction for the branch. But for pi-SDE, we can clearly see this branch from one in this direction, one and another in another direction, which is consistent with the biological study from the experimental study. Also, for the branch 2, if you look at the sugar net, they even have opposite direction. It's opposite direction along the time development process, which means that the trajectory net cannot, even they impose the principle of least action, it's still, because it's deterministic, it cannot predict the correct direction. But for present and pi-SDE, we both can predict a correct direction along the time point. And here is another example is from the blood development process. It's connected at 3 data point, and this figure shows how this data from different distribution of the data the cells at a different time point. And also, as previously, we can also reconstruct the potential landscape. We found that our method can give a very can provide a biologically reasonable potential, which shows a progressive dynamic process along the part. But for preset and they also largely dominated by the last day point, day 6. And if we also look at the cell velocity in this data, we can find that for present, most of the direction are misleading to some minor branch point. But for our method, the direction we predict is consistent with the that the cells are majorly divided into this two branch point, shows that we can more clearly delineate the developer dynamic process of the cell development. Also, since it's a generative model, we can use the learning the model to make some perturbation in silico perturbation test to tell us to introduce some perturbation on the different genes to check their treatment effect and to predict the treatment impact. We have did some in silico experiment to perturbate some important genes that are related to the different branches, and we can find that if we predict important genes, they have the method can correctly predict their cell fate development process. But if we perturbate some non-important genes, which is they have little effect on this treatment effect. So it indicates that with this kind of machine learning method, we can use it to make some in silico test to facilitate to make some discovery from the experiment. Finally, before I conclude, I want to say that our method, by introducing some physics information, can greatly help us to learning the Darling process by stabilizing the learning process. For example, here the horizon picture shows that the different learning rate that used in the neural network, and here vertical figure shows the different regularization strengths. If lambda equal to 0, there's no regularization term in this model, and we can find that if we introduce less regularization term, we can find that the learning process is not stable, because you can see that this shows the loss function is not stable, cannot stable. But when lambda, when you consider less physical information, and when we introduce more and more physical information, and we can stabilize this learning process and achieve very good performance in the loss. For example, here we have they have a loss, no noise, no loss, and but if we less regulation term introduced, they also have high loss. So it means that HJ regularization can gather the PSD to find optimal potential and then antigen landscape with accurate predictions. The basic idea is that if we do not impose any regularization, the search space is very huge, and it cannot be stabilized. But when we introduce this kind of introduce the regularization, you can reduce our searching space to help the machine learning algorithm to find the more realistic solution and reduce the searching space. So here's the summary. Here in my talk, I show that we can combine the optimal transport physical machine mechanism and diffusion model to provide powerful tools to analyze complex biological data. And here we introduce a coarse-grained method called GraphIP. It works on the graph, and also we introduce some physics informed neural network method. It's a fine-grained method. It encodes the physics principle into the loss function. Both methods can have a high interpretability, both in the potential landscape or cell-cell interaction or cell velocity. Also, the PSD can also be used to do some in silico perturbation of the genes and to help us to find some new findings, and also can stabilize the training process. And here is a reference of this paper. If you are interested, you can find more details about method. And currently, we are working on the method to develop some to modeling spatial temporal dynamics. Recently, the development of the spatial omics can not only provide the time information, but also the spatial information of the cells at each time point. So how to integrate this kind of spatial temporal data is still very challenging. The problem is still we lack the cell-cell correspondence between the cells. And we recently developed some method called a graph sport to align the different slides or spatial slides with the optimal transport. But how to modeling and prediction learning the dynamics is still challenging there. So finally, I thank my collaborator Suo Zhang, and most of the work is done by my graduate student Qi Jiang. And thank you very much for your attention.
Yeah, thank you very much. Thank you. Any questions?
I have a question about the data sets there you're using.
Okay.
Right. Hi, I'm Tom Berlin, research software engineer, UMass Lowell.
Data sets?
The data sets with so many 40,000 cells or 40,000 cell states or something like that.
Yeah. Yeah.
For example, is this from one individual over 8 days? Or are they scattered over many individuals?
It's a different individual. For example, if it's connected from a rat, you at day one, you will kill this. So, for example, for the mouse, you will kill them and collect the samples. And day two, it's for this one is in virtual. For this one, it's from different mouse.
Is it no more than one data point per individual then? I'm just actually
maybe more than one individual. They will have because as the early development states, you have very less cell can collect it, and you usually can maybe more than one mouse.
So is it 49,000 individual mice or
49 cells from okay. Maybe more than 3 mice.
Okay. One time point per mouse, but fewer than that.
One time per mouse, yeah. Because after you collect this data, the cell, the mouse are killed.
Okay. All right. Thank you.
I also have a follow-up question, if it's okay. So since we know that this single-cell RNA-seq is containing a lot of technical variations, dropouts, and batch effects, assuming that a data collected at time t1 has the same prior information with another data collected at t0, is it true? I mean, since they will have some batch effects, right? Because they are collected from different sources, different animals. And maybe, I mean, the data sets coming from different time points might be representing a little bit biologically different information based on these cells.
Yeah. It's a difficult question because it's a good question, yes, and they do have the batch effect and such as technical effects. And here we mainly follow the standard procedure to analyze this kind of data to do some normalization and batch effect removal. But yes, it's still hard to disentangle this real biology dynamic part and the batch effect part. It's a problem. And we also recently working on some develop some causal inference based method to try to disentangle this different source of variation in this kind of data. But basically, in this model, we assume that this batch effect have been corrected, and technically you can have correct, and we just need to focus on the modeling dynamic part.
Okay, thank you.
Thank you.
All right, thanks. Other questions?
Yeah, just how a few questions. Just thank you so much for presentations. And so this method is still like the supervised. So when we processing data set, we still like do we need to label data set or for each cells, or we don't need to label?
Yeah, for the PySDE is unsupervised. We do not need the label of the cells. And for the graph FP, you can either have you need to first clustering this data into clusters, or you need to label this cell. But the label is we use this label to reduce the state space into finite space to reduce the computation.
Okay. Okay. Yeah. Thank you.
But the label information is not used during the dynamic part.
Okay. Yeah. Thank you so much.
Thank you.
Other questions? Yeah, maybe I have a quick question as well. I have a student actually who's also doing the experiments on mice, and then the goal is to with the few data points, and the goal is to detect cancer. So basically, do you think that there is a way to detect critical transitions using this kind of methods when there is like a sudden change, or a patient is healthy and suddenly becomes sick? So how do you see? I mean, do you see that this could be useful in this kind of context?
Yeah, I mean, I understand you want to identify some critical transition point. Yeah.
Yeah, exactly. Yeah.
I think some method use some from Lin Wan, they develop some entropy based network entropy to detect or dynamic network biomarker to quantify this cell their states. They are basically from the dynamic info at the transition data point. This cell should have a large variation, and they have a weak connection within this group, and they have some method to based on the dynamic systems.
Okay. Yeah. So this is what we are trying to work on, but it's nice actually that you already so you have some references. Yeah. So maybe we'll discuss offline with the student. Yeah. If you can send me the references, that would be nice. Yeah. Thank you.
Okay.
Yeah. All right. Okay. Thanks a lot. Any other questions?
Here, I have one last question. Sorry for keeping things
No, it's
long.
okay. No, go ahead. Yeah, it's open. Yeah. Thank you.
Based on these sketches, bar plots that you provided here, in this the previous slide, yeah.
The previous
the thing that we have all these sorry, no, these bar plots and
second part or first part?
Okay, in general, let me ask you, do you here, for example, no, just show me some images. Do you expect to have the same type of cells or the same number of clusters for predicting this transition? And does the size of the populations matter for your predictions?
Yeah, you mean the different time point have different cell number, right?
yeah, does this cell number mismatches affect, and does
Yeah.
the size of the population affect? Because during the transition, you might not see the same amount of cells in another time point because they are simply
Yeah.
differentiating from
Yeah.
one state to another.
Yeah.
Will it affect, you know?
Yeah, yeah, I understand. For the cell development process, some cells may be growth and some cells may be death. And yes, there are fields of work try to estimate the cell growth rate and integrate into their method. For example, even for this one, yeah, for this one, for PreSat, they have two parts, and one is that they do not consider the cell proliferation, and this method they also can consider the cell proliferation, and basically we found that the method we theoretically they should be more general, but in reality, we found that the model without the modeling this part have good performance. You can see that. Yeah, you can say that because the problem is that if we want to estimate the cell growth rate, it's very hard problem. It cannot. You should impose some understanding whether there is understanding, but this can be very not accurate to estimate this growth rate. But from the data we can find, if we do not use the growth rate, the method also have a very high accuracy. It's a difficult problem to accurately estimate this growth rate G.
Okay, thank you.
All right. Any last questions? Okay, seems not. Okay, thanks again, Lin, for the nice talk, and then we'll keep in touch by offline because I just want to carry our discussion. Okay, thank you again for everyone, and see you next week for the next seminars. Yeah, thanks. Bye bye.
Thank you.
Bye.
Bye.